So welcome to today's signature lecture series from the ARC Center of Excellence for Eco-Efficient Beneficiation of Minerals. We're lucky to have Pablo Brito Parada from Imperial College. Today, I'd like to start by acknowledging the traditional custodians of country throughout Australia and their connection to land, sea, and community. And we pay our respects to their elders, past, present, and emerging, and extend that respect to all Aboriginal and Torres Strait Islander people today. And so Pablo is going to talk to us about something which sounds very interesting, tracking particles using positron emission particle tracking, and to see where they go from the pulp to the froth, or back or forth, or where they go. So with that, it'd be great to hear what Pablo has to say. Thank you.
Josh, thanks very much. And yeah, first of all, thanks for the invitation to deliver this signature lecture. It's an honor to do so and to be talking to you all this morning, afternoon, whatever time it is for you. So yeah, this talk indeed will focus on a technique called Positron Emission Particle Tracking, or PEPT, which has great potential for studying and better understanding flotation systems. And the idea here is that throughout the talk, I will, of course, explain how the technique works and some of the applications that we have found for it in flotation, for mechanical cells, for fluidized flotation cells, novel designs, to study retrofit designs and operating conditions, while also focusing and explaining a bit what's the role of the tracers and what we can do at the moment, and what are the challenges that we still have to keep exploiting this particular technique. Okay, so let me start by showing you where we're based. So the Advanced Mineral Processing Research Group is part of Imperial College, the Department of Earth Science and Engineering. And this, if you haven't had the chance to visit us, is part of our South Kensington campus. So what you see in the middle is not the whole area. It's really compact, actually. What you see in the middle is the Queen's Tower, and the building around that is the South Kensington campus for Imperial. We're surrounded by fantastic museums in this part of London, and also Hyde Park that you can see here at the top. So yeah, if you haven't had a chance, please come and visit us at some point. And in particular, this on the right is the Royal School of Mines. So we have a long history of resources, mining, processing, and metallurgy. And by the way, this year in particular is very important for us. Is the 175th anniversary of the Royal School of Mines. So there's a whole program of lectures and events around this. And this is part of the team in one of our campaigns in Spain recently. Now, just before delving into the topic, I thought of showing a bit of what we do in other areas as well. Historically, the team at Imperial has worked a lot on flotation, but we also work on other areas of mineral processing, leaching, rapid separation. The way that we tend to approach these problems is to start with the physics of the problem. So we work a lot more on the physics than the chemistry. So focus on the physics, novel characterization techniques, measurement techniques such as PEPT, the one that I'm going to be focusing on today, and then combining it with simulations to then start using this for equipment design, optimization at the lab scale, and taking it all the way to implementation on site. And on the way, developing with the models also control strategies and looking at the bigger picture, for example, material energy nexus, the role of water in mining and processing, and so on. But of course, the topic today is going to be flotation, and I always like to show first or talk a bit about how flotation affects across the different scales. And most of you are, of course, very familiar and aware of this, but we have interactions at the molecular level, of course, surface phenomena, and then we go to particles that are micron size that interact with bubbles that can be half millimeter, a few millimeters in size. So it becomes very complex when we start dealing with all those different scales. And of course, at the end, we have flotation cells that can be, at least for conventional cells, hundreds of cubic meters in size, the largest being around 600 cubic meters. So how do we start understanding, in particular from the area that is of most interest to me, what is the effect of hydrodynamics in flotation cells, and how can we understand it? And of course, I think that the only way to optimize flotation systems is to focus on the fundamentals, get that fundamental understanding, of the different phenomena taking place in the cells. And then flotation is, of course, complex. Conceptually, it's very simple, of course, but it has a number of complexities. It's a multi-phase system. It's opaque, and I will come back to this point that is highlighted here. It's polydispersed. We have size distribution for the particles, for the bubbles. It's turbulent in the pulp. We have the froth phase that has its own complexities, and it is very interesting and at the same time very complex to understand. And on one side, we have the hydrodynamics, the froth stability at the top, mineral bubble interactions. All these are really key to understand what's going on. But again, the system is opaque. So if we're interested in understanding what is the behavior of particles in the system, other techniques that can be used that are non-invasive, that can be used to understand what a particle is doing in the system, don't really work. If we think about PIV, particle image velocimetry, then rely on the system not being opaque. You need to use a laser to then be able to track seeding particles that are used. And while you can study flotation devices using PIV, it tends to be very low concentrations of solids, if at all, other than the particles used for the seeding. And that sometimes makes it a bit more complicated. Two-phase systems don't really behave as three-phase mineralized systems, so that poses a challenge. So the question here is, how do we track this motion of particles within a flotation cell? How do we can do that? And why we want to do that? Well, again, we want to do it because if we can understand what hydrophobic, valuable particles, valuable minerals are doing in the cell, and we can understand what's the behavior of the gangue particles as well, then we can compare different conditions, start optimizing. We can also compare different designs or retrofit designs for the flotation cells and enhance performance, or we can study novel cells as well. So that's the point of this talk. So introducing positron emission particle tracking, or PEPT, as a technique that can be used to track particles within a flotation system. So what PEPT allows is exactly that, the visualization of the path that the particles will follow within a system. And those particles for PEPT are radiolabelled tracer particles that the trajectory of which can be measured using a positron emission tomography. So that's exactly the PET scanner that you will find in a hospital, as you can see on the left. And what you can see on the right is the array of buckets and sensors that are used in a PET scanner. And of course, in a PET scanner, the tracer that is used, that is radiolabeled, tends to be an aqueous solution. So that solution is injected into a patient, and then therefore we can get 3D images of the flow of that tracer to better understand from a medical point of view what's going on. So the idea here is to take out the patient and then instead replace the patient for a flotation cell. And that's what is being shown here on the left. So PEPT, instead of using that tracer, uses a particle as the tracer. The technique to do this was developed initially at the University of Birmingham by Professor David Parker in the early 90s, and it slowly made its way into mineral processing applications, initially particularly for comminution, particularly at UCT. And at some point also it was used for flotation. My colleague Jan Cilliers initially used this technique in a Denver cell to track what the particle was doing. But for batch flotation cell, there is a complication because you get the tracer at some point and then how do you put it back? And I'll get to that a bit later in the talk. The technique is very useful, again, if you can have a flotation device or any device that can fit within the given PET scanner. And the one that I'm showing here is the one at Cape Town, Itemba Labs in South Africa, which was originally a PET scanner from a hospital in London that a group took to Cape Town to contribute to PET research. Now, what PEPT does, and what is being shown here on the right as well, is that tracking of the particles. What you can see here are those buckets of sensors that we saw on the previous image for the PET scanner. And eventually what we want to get is the trajectory of a given particle, ideally in a system that is otherwise the same as a flotation system. So height solid constants, that doesn't matter because it doesn't rely on an optical technique to do this. And those tracers can be 100 microns to several millimeters in size. So we had a wide range in which we can produce tracers and apply it to flotation research. Now, how does this look like? This is the sort of trackings that we can get for flotation. And here you're seeing a hydrophilic particle track that is a particle that is a valuable mineral. And you can see that eventually this green line here represents the interface. You can see that the particle travels happily in the pulp phase, sometimes enters the froth. There is drawback events, which is really interesting to see. Then the particle goes into the froth, starts moving slowly, probably either attaching to the lamella in the froth, or perhaps just traveling as a trained particle, depending on the velocities. Is very tricky still to determine that difference. It's something that we keep working on, but you clearly see that it goes into the froth and it eventually makes its way and overflows the cell. So I always find this fascinating to see. Every time we do experiment, the tracks are fascinating. But it's not necessarily the most useful information in terms of comparisons and quantifications. So what we really need to do, and what becomes very useful, is to then start taking half an hour of PEPT measurements, and then averaging that data, and then plotting these as slices. In this case, for example, the average tracer speed, comparing two different flotation cells, in here on the left without any modifications, and on the right using a retrofit design that sits at the interface. And this already starts showing really interesting things. So having that retrofit design in this dashed red line or rectangle here shows that the loops that you see in these streamlines here are actually compressed in the flotation cell. You can see one loop here, one another here, that is typical from these type of stir systems. The retrofit design not only compresses those loops, but what we can see is that the tracer particle, the valuable mineral, has a clearer path towards the interface, and then that eventually helps the reporting to the interface. So for these systems, we can now not only measure or estimate recovery and grade of the concentrate, but also understand what the valuable minerals were doing using a representative tracer. And that aspect is very important. What is a representative tracer? And I'll discuss briefly how tracers can be produced for PEPT and what we have found to be really useful for these studies. But again, going from designing a flotation cell that can give us representative behavior of industrial flotation, all the way to obtaining really quantified phenomena within the flotation cell. And again, what we can do is not only having a hydrophobic particle, but also studying hydrophilic particles. What is the gang doing? As you would expect, for the hydrophobic, as we saw before, we can see the particle going into the froth phase. For hydrophilic of gang, you wouldn't expect that to happen, or at least not that often, unless the particle isn't trained. If we are focusing on relatively coarse tracers, then it's very rare to see entrainment for the hydrophilic. We'll need to go to very finer particle tracers to be able to see that. But still, being able to understand where in the cell the particle or the gang particle spends more of the time becomes really, really interesting when it comes to operating conditions and also to design. Now, stepping back a bit just to try and describe how PEPT works. As I say, the key is the tracer to start with. So what we have is a particle that is labeled with a radionuclide, and that radionuclide decays by emitting positrons. So the buckets and sensors, so the sensors in the PET scanner, will record when those positrons annihilate. And when that happens, then they release almost back-to-back, almost linear gamma ray photons. And that is what that gamma ray photons is what the sensors can detect. So that's what I'm trying to show here. These images, we have the tracer, positron annihilation, and then the detector heads detect those gamma rays that particle emits. And then we can reconstruct the trajectory because we know that the sensors on opposite sides detect those almost linear back-to-back gamma rays. Now that happens very, very often, really quickly. And what happens then is that we can then, by having multiple events, we can use triangulation algorithms to identify where exactly that particle was. So that algorithm allows us to determine the line of response, and therefore we do that over time, and we can have an exact position of the tracer over time. So that is very useful, and that's how the technique works. We have a tracer, we have a technique, and there is a lot of work that has happening and still happening in terms of refining those algorithms and analyzing the data that comes from PEPT. There's plenty to do. I'll discuss that a bit at the end. The other side of this research is, of course, the scanners, the hardware that we use. For PEPT, any PET scanner can be used. So we can talk about, for example, the one that I showed before for South Africa. That's a Siemens ECAT PET scanner, and that's the one that we have used the most in our flotation research. And you can see here the cell. At Itemba, there is also a ADAC vertex model, which can be useful because that one has parallel heads. So in the Siemens ECAT that you see here, we're by the nature of the design, we're restricted by the circumference of the PET scanner. Typically, the patient lays down, and there you can position the patient to be within the sensors area, but that limits us quite a lot in terms of flotation. With the parallel heads, then that helps a bit with the problem, if you can still fit it within some restrictions here. Now, at Birmingham, where the technique was developed, there's been, over the last few years, some developments as well. There is a particular array of sensors that they call Super PEPT, better resolution. It has some flexibility in the way that you can position the sensors. You're still limited to a circumference, which is 40 centimeters, but you can rotate it. So you can have it horizontally or vertically aligned, which has been pretty useful. There is also microPEPT, which we haven't used yet. Smaller diameter for the sensors, high resolution for very, very small. So this is not dissimilar to the sort of PET scanners that will be used for animals, for small animals, in that field. But again, the smaller arrays allow you to get better resolution as well. And one that I will be talking about later in this talk is the large modular array, or LAMA, which we tested a couple of years ago in Birmingham. In this case, we take the buckets, and then we can arrange the buckets and the sensors in whichever way we want. And that has been really useful for taller flotation cells, non-conventional or not mechanical flotation cells, but fluidized flotation machines. And I will show and discuss this application later in this talk. Much more to do as well. There are challenges when it comes to the size of the flotation cells that we would like to try, but particularly things like the modular array are opening the door to a lot more potential applications for flotation. Now, in terms of the tracers, I already mentioned that we need particles that are radiolabelled. But what do we want from these particles? On one hand, we want them to have high activity. High activity means that there will be a high event rate, and therefore we'll have better temporal resolution as well, and spatial resolution for the identification of particles. We want high radiochemical purity. There is noise that other species present might have, so that noise might lead to inaccurate line of responses in the data that we analyze. So it's very important to have radiochemical purity. And very important, as we have found out over the number of campaigns that we've done for PEPT, is tracer stability, depending on the technique that we use, of course. But for some of the tracers that we use, the tracer can leach. If that starts happening, then we have, of course, activity, radioactivity in various parts of the cell, and therefore we lose the signal. There is too much noise, and therefore we cannot really track the particle because the particle is no longer the one that we have designed. Something important to mention at this point when talking about radioactivity and exposure is that, for example, working on a week and a half campaign using PEPT, measuring 2-3 experiments a day, for example, the exposure will be less than what you get from an international flight. So it's a relatively, of course, you need to have precautions. We need to have really good protocols in place to produce the tracers and to deal with the tracers. But the level of exposure for a well-planned activity is not gigantic. Now, how do we get that radio labeling to happen. There are effectively two ways of doing this. One is direct activation. In this particular technique, what we do, and it's shown here as an schematic taken from this paper from a colleague in Birmingham. In direct activation, you convert an existing atom into a positron emitting particle. And we do this by irradiating it with an ion beam. So there is a cyclotron, for example, at Birmingham or at Temba, and that is used to do direct activation of a tracer. We have done that for chalcopyrite particles, for example, and then we can use the exact mineral to be able to act as a tracer. There are also indirect activation methods, and we have found those to be very useful, and those are the ones that we have used for most of our work in Cape Town at Itemba Labs. So ion exchange, and again, shown in the schematic, what it does is replace a radioactively inert atom with a positron emitter one. And then we have, with this allows to use it as tracer. Or we can do surface labeling, in which we just make sure that those positron emitters can be attached to the surface of other atoms. So that's the surface labeling technique. I will talk a bit more about indirect activation in particular, because the type of surface modification that we have done to develop but valuable hydrophobic or hydrophilic tracers depends a lot on this technique, and also some of the recent developments as well. But let's start with some of the examples. And I want to show in this talk primarily two types of cells, our small recirculating bench scale flotation cell and a fluidized bed later on. Now, as I mentioned before, a bench scale is what we need, because in most cases we're limited in terms of the size of the equipment that we can use for the PET scanner. But the other important thing to highlight here is that the flotation cell that we use recirculates the concentrate, and that allows us to run continuously. The way that we have designed the cell and the systems that we use is that the behavior of the froth is that we would expect at industrial scale as well, because a lot of the work that we do is based on froth stability. So we're very interested that that particular bench scale cell replicates the behavior from that point of view. It also becomes really useful because let's say, if we were running batch flotation cells in a Denver cell for PEPT, every time the particle would overflow, we would have to find the particle and then put it back into the cell to measure again. So again, some of the first studies did that, and it was really good for developing the technique. But we quickly move into a system that allows to run continuously. That really allows to get enough data for those averaging values that I showed before. So this is what the flotation cell looks like. It is relatively small, 4 liters, 4.5 liters. It overflows, we have the launder, and then we recirculate either with a sump or directly into the pulp. This allows to get a steady state and therefore a stable system. That fits nicely, as I showed before, into the camera of PET scanner at Itemba. And the type of tracers that we have used there, as I mentioned, are indirectly activated tracers. Now, the technique here, what we do is using a resin and get gallium-68 to develop the tracer for PET. The challenge, or one of the aspects to consider when we do this, is that we start with the resin, and then we need to consider how to make it a representative particle for hydrophobic or hydrophilic minerals. The way that we do this for hydrophilic minerals is relatively easy. We have the resin, that is already the tracer, but we cover it with an epoxy at the surface that has hydrophilic properties. So that's already relatively easy. We have a hydrophilic or a gang particle. What we have done typically for this to get the hydrophobic tracer is to use even finer Ballotini beads, silanized Ballotini beads, that therefore have hydrophobic properties. This allows to get tracers that are, in this particular case, I'm showing 500 microns, so coarse, but we have also come down to 250 microns as well using this surface coating technique. And we have enough activity in the tracer to have really good results. Now, something that we have done a few years ago is also to get tracers that are more representative of the water within the system. So at this point, we had done a lot of work on hydrophobic and hydrophilic tracers, and that tells you the behavior of the particles. But we were trying to understand as well what was happening to the liquid within the cell at conditions that were representative. So again, when running at high solid content in the flotation cell. So what we did is to develop an alginate hydrogel that could be used as a tracer, with a caveat that it was a coarser particle, but it was neutrally buoyant. So at least it started giving us an indication of what the liquid was doing within the filtration cell, which was really interesting in terms of inferring what the results meant for entrainment, for example. And that is what I'm going to be showing here. So from hydrophobic, hydrophilic in the center, and then the alginate tracer on the right, same conditions in the cell, and starting with the tracks to see what happened with the system. As you would expect, again, you see nice tracks going into the froth for the hydrophobic or valuable mineral, primarily staining the pulp for the hydrophilic. And then, again, you cannot really compare from just a few seconds of tracks, but on the right, this is what the alginate does. As I mentioned before, while it's always fascinating to look at this, it is really when we start getting the averages that we get valuable quantified information. The way that we do this is by generating an Eulerian visualization. We take the boxes that we're considering for the measurements, and then we take slices throughout the tank. We tend to avoid, for example, baffles. In that particular design, we have baffles in the cell, so we avoided those. And then we take half an hour or an hour of measurements and can generate these 2D Eulerian plots, in which we can plot the average streamlines for the particles, but also any other variable of interest. In here, for example, what I'm showing is the occupancy in the flotation cell. There are various ways in which we can define occupancy. They vary slightly in the way that the results are shown. This particular one shown here is the number of times that the tracer passes through each voxel relative to all the passing events in a given slice. So we can quantify that. We can see first the streamlines. We can see for the hydrophobic, of course, we can see clear streamlines going throughout the froth and all the way to the concentrate. Less so for the hydrophilic. The streamlines show some entrainment events. You can see clearly this particle here is being entrained at some point, but most of the movement is downwards, as you would expect, as drainage happens as well. We see that less often for the alginate. And for the occupancy, then you can clearly see that the values for hydrophobic and alginate in the froth section at the top are basically non-existent. So there are very, very few passes that give these average streamlines. But really, while for the hydrophobic or valuable particle, we can see a lot of occupancy, higher values near the pulp-froth interface, and then in the froth, what we see for the hydrophilic and the alginate is a lot of time spent for the hydrophilic below the impeller in particular, a bit less so for the alginate. But overall behaving similarly, which is what we expect. Those hydrophilic particles, particularly in the highly turbulent region near the impeller, are expected to behave quite close to the liquid. They will follow the liquid. And of course, there will be considerations in terms of, because of the density and drag forces and so on, we won't expect it to be exactly the same, but quite close. So that's what we're seeing here. So now we can quantify those passes and start understanding what's going on. Also, what is useful once we have this information in this format that has been quantified is to start looking at the difference in these values. And that's what this particular plot shows here, a comparison between the alginate and the hydrophilic. Are they really different, and where exactly in the cell is that we're seeing the differences? And this was really interesting because what we saw is that the occupancy of the alginate tracer was almost twice that of the hydrophilic particle, particularly at the pulp-froth interface, which is what we would expect as well. We see that liquid behaving particle, the alginate, getting closer to the interface. We don't see much entrainment. Again, remember that this is a relatively coarse alginate particle, so entrainment would be difficult, but we see that behavior going all the way to the interface. And in the same way that we plot occupancy, we can also plot velocity. And what we were showing here in this particular publication in Chemical Science was the effect or the behavior that we saw at two key parts of the flotation cell. So at the level of the impeller, which is the black box here on the bottom left, and also at the interface in green here. So we're plotting here those vertical velocities for the three different particle types at those two regions. I won't go too much into the details here, but the important thing is that we can quantify and compare. We expect high velocities near the impeller. We see some similarities between hydrophobic and alginate compared to the hydrophobic particle, not as much, of course, as once we get to the interface, which is when the vertical velocity for the hydrophobic will be higher and ascending as it goes through the froth. But we can quantify. Something that is not shown here and is part of separate work is what happened when we start changing operating conditions and also changes in design. But as a proof of concept that velocities could be plotted and assessed. But again, what's that difference in velocity? How much different is our hydrophilic tracer to our hydrogel or alginate tracer? We can plot vertical velocity difference or the relative change, and we start having a look at some of the information here. What I found more useful in this particular case is to use the Jensen-Shannon distance as a way of comparing this. And the Jensen-Shannon distance, or GS distance, is effectively a non-parametric statistical test. In the scale that you see here, I'm showing from 0 to 0.5, but if what happened when we plot this is that 0 would mean that the distributions are identical, whereas the higher the value, all the way to 1, would be completely different. Here, the highest values that we get are 0.5, particularly close to the interface and in some regions near the impeller. Again, just for one condition. As we change things for impeller speed, for the sign of the cell, or aeration, this will also change as well. But this has been proven to be a really useful way of looking at those differences once we have all this data. Because the problem then is especially that, data analytics. How do we start making sense of the massive amount of data and information that we get? Now, I have mentioned flotation cell design already, and it's something that we have been doing a lot of work on over the last few years. In the previous slides, you have seen that we were using a Rushton turbine. That was the first design that we used because we were trying to compare against well-understood hydrodynamics in chemical engineering. Eventually, we moved to a rotor-stator system more representative of flotation cells. And at that point, we could, for example, say, okay, what happened in a flotation cell if we don't have the stator? If we only have the rotor but not the stator, what happened to the flow patterns here and what happened to the particle tracks as well? In here, for the valuable mineral and the hydrophobic tracers, you can see exactly what happens. Those loops that we typically see in rotor systems actually change once we have the stator. We're concentrating a lot of the energy around the impeller, constrained by the stator. And that has some advantages as well, but we can, again, quantify them and understand them. Again, for the hydrophobic tracer, but then trying to get a difference in velocity or understand the difference in velocity for different tracers. Again, using Jensen-Shannon distance to do that, understanding where those differences are. Now, going further, is typically we know that rotor-stator systems work well. One of the main problems that we wanted to tackle initially with PEPT was what happens at the interface and when the interface can completely destabilize the froth. So one of the retrofit designs that we tested, and the one that I showed with one of the first slides actually, was the use of a mesh that sits at the froth, the pulp froth interface, with the idea of reducing any turbulence and allowing the recovery of coarser particles or stabilizing the system as well for finer particles. And we have tested a variety of these. In this particular one is a honeycomb mesh design that can be seen here at the bottom, and again, sits at or just below the interface. In this case, we tested this just with the rotor and mesh, adding the stator as well, and then everything rotor stator and also the mesh. you can clearly see how the streamlines, particularly in the froth phase towards the overflowing lip, start changing completely. They're not that clear, and the overflow is not that clear from the streamlines just using a rotor, all the way to everything else in the system that has really clear streamlines, even at the center of the tank, which is not necessarily the same for just using the mesh. Again, this has been really interesting, really useful for then, in this case, 3D printing novel designs for the meshes or different stator systems to optimize the design and then compare at a larger scale. Now, I also mentioned that we have used PEPT for novel flotation technologies, and in this case, for a fluidized flotation cell. In particular, for this study, the coarse air, a large lab scale coarse air fluidized bed cell. And of course, a lot of you will be familiar with the design. The lamella plates at the top. In this particular case, we use a coarse tracer. We were interested in coarse particle flotation. That's the whole point of this technology. The machine is highly selective, and the lab scale is more than 2 meters tall. So again, when it comes to PEPT, we were never going to be testing this at Itemba Labs because of the restrictions in size, but we could do that using the large modular array that I show in Birmingham. One of the key points for this, of course, was also to get the system to run continuously for as long as possible, but it did present a lot of challenges in terms of recovering the tracer every time to re-inject them because we couldn't get a recirculating system in place. Just to show a bit more information, these are some of the buckets in the large modular array, or llama. There is a high number of them. We have 48 buckets that we can position in wherever we want. In this case, at each side of the machine, and then following the angle of the lamella plates as well. So it's modular. It has an extended field of view as well, which allows to do this. A lot of cables, and this is how it looks in the lab. As I was saying before, on each side of the Corsair, we could have the buckets following this, the lamella section at the top, and then being able to capture as much information as we could from the system. In this particular case, we didn't use indirectly, but instead we used directly activated tracers. We tried chalcopyrite particles from 300 to 700 microns in size. So again, the challenge, if we went, in this case, we were focusing on coarse particles. If we want finer particles, the challenge remains for direct activation that the surface can change dramatically. And therefore, we stop having a representative hydrophobic particle. So we used chalcopyrite and silica beads for the hydrophilic gang as well, from 500 to 1,000. And again, pretty good level of activity. 2.5 megabecquerels and 6 megabecquerels for the silica beads. Also an important consideration here, and so far I haven't mentioned this, but most commonly we will use one tracer at a time. The system has a high solid content, gigantic number of particles, but then only one tracer. We can actually do more than one tracer at a time. And particularly when we have differences in activity, 2.5 to 6 megabecquerels here, what we did sometimes for some of the tests was having both tracers, hydrophobic and hydrophilic at the same time, and it just becomes an issue in the way that we analyze the data to differentiate those particles by their different activity. So that saves us some time. We can get more data for a given campaign time, and therefore maximizing the amount of information that we are getting. That's what it's called, or referred to as multiple particle tracking in PEPT. And again, as you would expect, the hydrophobic tracer actually was really great to see. It really quickly gets picked up by the bubbles and then goes to the lamella plates. Of course, that meant that every time we had to collect it and inject it again. But it was really, really interesting and valuable data. Whereas the hydrophilic, we see a lot of the behavior going up and down, getting to the fluidized section of the cell, recirculation, and so on. But then we can start quantifying for different particle sizes what was the average velocity in different sections of the cell as well. We can get the tracks. Again, it was really interesting to see and try to understand for this particular size of cell at the lab scale, what was the behavior particular at the lamella. But it was pretty much straightforward, but a really good of concept to then test different conditions and changes to these designs. And again, focusing a bit more on the tracks at the lamella, in this case for vertical velocities at different points. It was interesting because it was, in this particular case, primarily in some of those sections that we could see most of the tracks. We can then also get time average vertical velocity, occupancy for these, in this case, in terms of number of passes as well, and start quantifying. Now, the last bit that I would like to mention is something that is more recent, and a more recent study was actually presented by someone from the team at the previous flotation conferences, and that's the use of novel tracers that allow us to assess partial liberation. So far, for the indirectly activated tracers that I showed, we either have those silanized silica in the entire tracer, giving us a fully liberated valuable mineral or a representation of one, or a particle that represented 100% gangue. Of course, liberation is very important in flotation, as we all know, but the challenge was, particularly for indirectly activated tracers, how do we construct a tracer that allows to study this? So with help with colleagues at Itemba and Justifysics, we developed this. So we had a new tracer that emulates partial liberation. We have to use a mold for the resin. So we have that epoxy-coated tracer, which at this point is hydrophilic. We have it in the mold. And then we have the silanized Ballatini only in the sections that we're interested to cover. That allows, for example, to get a partially hydrophobic tracer, only covering half of the system. Of course, this is still pretty high in terms of liberation, but as a first step was fantastic to be able to get. So we were able to test 50%, 25%, and 75% liberation using this technique. In this schematic, it looks simple. In reality, getting this technique, the development of it, and then getting the coverage right was complex, of course. And I'll focus here particularly on 25 and 75 because the other tracer had some issues during operation. For you to have an idea of the system, solid particles, glass beads. So we had a representative system using only glass beads and then tracers with different properties. Coarse particles ranging from -200 to +100, middlings and a 1:1 mass ratio. Almost 1 centimeter per second for JG, and you can see the details for the reagent system here as well. Again, recirculating in the cell so that we can run continuously for a long time. And this was really interesting to see because in terms of the overall streamlines and velocities and occupancy, streamlines in particular, we can see that there is not a gigantic change, at least not here in terms of the streamlines. Both of them show those typical loops that we would expect. But with the 75% liberated tracer, what we found is that it spends longer near the froth and the lip as well, which you can see here. So we can start seeing at least a small change in occupancy, in the velocities, and in the reporting to the concentrate, which we also measure separately. So again, as a proof of concept, we can now develop those more complex tracers that allow us to study more things. Something that we also start doing when analyzing this data is start looking at all the PEPT data in a different way, not only the tracks and not only the averages, but also started studying the cell considering the different sections. So in this particular case, we divided the pulp into lower section and upper section. We vaguely define the interface or the area or the volume around the interface, and also 2 sections for the froth. Now, this, I think, becomes even more interesting because you can understand what's happening with the tracers, but in the really turbulent zone, but also at the interface and the froth. What we see is that the residence time this redistributes upwards. We have more liberated tracers that spend more time in the upper froth, but again, we can quantify it, 14 versus 7%, which is something that we haven't done in the past. We can also see reduced recirculation. The liberation of the particle reduces the time in the upper pulp because basically it doesn't get there that as frequently as the higher liberated particle. And again, we can quantify it. We see a change from 31% to 24% in the upper pulp, and then also reduction at the interface, as you can see from these plots. And something that I also think is quite good in terms of visualization is the use of Sankey diagrams that are typically used for material flow analysis, but to understand how much time, or what's the percentage of time that the particle or the tracer spends in these different regions. So again, here you can see a lot of time spent in the lower pulp, upper pulp, and then we can see exactly what happened at the interface. Less particles goes to the lower froth, and then less to the upper froth and overflowing. So again, excellent tool for visualization, but also for comparing and getting more information from these tracers. We can also start studying residence time distributions for the cell, comparing 25% liberation to 75% liberation. We see, again, better kinetics from that point of view. In the orange one, that is the 75%. We see more passes and therefore better performance as well. Something that this implies is that increase in liberation from 25 to 75% reduces back mixing and also the mean residence time in the cell. The tracer just goes through the system more quickly, as we hope would be the case with higher liberation. So to conclude, hopefully I have managed to convey that excitement about how PEPT is really powerful technique to understand key phenomena happening in flotation cells, both in the pulp phase and the froth phase. That is a fantastic way of optimizing flotation hydrodynamics by first understanding what's happening and then either changing operating conditions and optimizing, assessing retrofit designs for existing cells or completely new cells. Also the fact that, of course, this relies on advancement that have happened particularly in hardware and different PET scanners and technologies. This has been essential to test novel flotation designs like the Corsair. Also that working on tracer development has been instrumental to the studies that we have carried out. Without that expertise for different applications, it would be impossible to get more meaningful data for tracers that are representative of flotation. And of course, there are still several really interesting areas for development here. For example, in data analysis for the tracers, something that we have been working on is applying machine learning, but in particular also Bayesian techniques to be able to get more meaningful and better and more quickly data from these experimental campaigns. And of course, I would like to acknowledge this is not just what I've been doing, but the whole team, particularly Diego Mesa, Katie Gold, Stephen Neitling, Jan Silius, and Mike Van Herden, and Ray Perrin. So yeah, thanks to all of them for exciting and valuable contributions to this presentation. So with that, I would just like to say thank you, and I'm not sure I'll be happy to answer any questions if there are some, and we have time.
Thank you. Fantastic talk. Really, really nice stuff to see. In addition to all of your expertise in mineral processing, you must have people very good at data analysis and algorithms to get all of that information out of the, especially when you're changing the arrangement of the scanners, right? I mean, yeah.
Every particular system has its complexities in the way that we get the raw data.
Yeah.
How do you convert that raw data into some more meaningful data that can be analyzed? And it's not over, but you're absolutely right. So people in the team and collaborators, both at Itemba and PEEK in Birmingham, have expertise on this. But we have also been developing our own algorithms to do the analysis over the last 10 years or so.
Yeah. Thanks. Good. We have time for a few questions. Kevin, you have a question?
Thank you very much, Pablo. Really enjoyed that. And yes, I think first time I heard about some of this work, we were also working on the Corsair, and one of the interesting conclusions that we reached was that there, particularly where in relatively small diameter units, there is a wall effect that relates to the ability of the gas bubbles to uniformly engage particles near the wall that are in the fluidized bed. And I think this is a brilliant technique to tease out those sorts of nuances.
And I think that's probably what you see when I was showing that some of the tracks in the lamella plates were particularly in the middle, not often on the walls. That's a really interesting point.
But I'm talking specifically about the wall around the lower fluidized bed.
Okay.
Yep.
Oh, I see. Yep.
Yeah. So if you're running it, I mean, if you're actually running a continuous steady state system, particles are moving in and through and down and out, and they could get out before they engage an air bubble. And if you consider the macroscopic scale of the air bubbles and the size of the particles, it's a particle that's near the wall, is seeing less of the air bubbles, and it's something that will disappear in a larger vessel.
Yeah.
Yeah. But it does lead to leakage in actual small scale systems that we often use.
One of the PhD students, Daniel Dodo, has a question. Thanks for the very interesting presentation. Do the tracers have a lifespan? How long can it be used for tracking? Can you reuse it for another flotation test?
Yeah, that's an excellent question. I didn't mention much on that. It depends on the activity. So that activity that I showed at the beginning, either 6 megabecquerels or 2.5, that will be going down over time. The tracers will have a half-time depending on the type of tracer or the type of technique that we use as well. The way that they are designed is that they decay relatively quickly, because you don't want to, at the end of the experiment, you have to put them apart until they fully decay, and they are no longer a hazard. With the tracers that we have used, typically we can run for about 3 to 4 hours, perhaps. Depending on the case, 4 hours might be a stretch, depending on the initial activity, but that's the usual length. So what we tend to do is to run continuously, and if we're testing different operating conditions, we keep recirculating the tracer for those 3 to 4 hours. Until activity is low enough that data becomes meaningless at that point. We still need to recover it in the end because there will be some activity. So we end the experiment and we start flushing the system with a meter that allows us to identify when it passes through, and then we put it apart until it decays. But yeah, that's a really good point. For the Corsair experiments was slightly different. We were not able to get a system that was recirculating the overflow on that system, so we manually find the tracer when it overflows, if it was the valuable mineral, and then we injected it back. But again, that particular one, we could run several experiments for 2-3 hours.
So Emmanuel said, can the technology be used in industry?
That's another really interesting point. The largest PEPT experiment for flotation today, it remains the one for the Corsair in terms of the 2 meters or close to that, because all other studies have been limited by the ring of the arrays of sensors of other PET scanners, whereas the large modular array gives you more flexibility to do that. In theory, you could have more of these modular array buckets if they were produced. So that's possible, although it's not trivial to get hold of these buckets and then do all the arrangements. Part of the limitation will always be size. So I wouldn't expect any time in the near future to have sensors or buckets in an industrial flotation cell. But the problem is not only that, the problem is also the sourcing of the particle tracers. So that's the reason why we don't do PEPT experiments in our labs. We have the cell. We could have a PET scanner in the lab, but we don't have access, for example, to the cyclotron to then get the tracers produced. whether it's directly activated or indirectly activated, you still need that source to get the tracers. So that would be the main limitation. Even if you were to have arrays of sensors at a larger scale, at industrial scale, the problem would be, how do you get the tracers? How do you collect them at industrial scale? I think that's why it's important that the systems that we use, the flotation cells that we use or design, are representative of what we want to measure and are representative of particle and froth behavior at industrial scale. So that's what we have spent a lot of time on making sure that that's the case, particularly for our smaller flotation cell. It would be fantastic if we could, but there are really, really difficult limitations to overcome.
So Emmanuel has a further question about what hydrodynamic parameters did you measure during the study?
Okay, so again, we have the tracks, we have the positions of the particle at every point during the time that we study. So we can get velocities, we can get occupancy values, and then based on that, you can measure as much as you want in terms of the hydrodynamics, as long as the resolution is enough. And that's something that we have also spent a lot of time on more recently, is on checking how much we can infer, especially once we have really high velocities near the impeller. To which point the data that we have allows to have representative values for hydrodynamics. But with positions and velocities, then you can determine as much as you want in terms of hydrodynamic behavior.
Yeah. Anyone else have questions or comments? So my question is, how much is it cost? Roughly 100,000, 1 million, 5 million?
What exactly?
Well, one of the medical ones or the
Mhm.
the whole modular stack for the Corsair, yeah.
Yeah, no, interesting. I would say that for example, the modular one, it's not that easy to source the hardware.
Yeah.
So it's almost invaluable. Getting hold of those is not an easy task. I'm not entirely sure what would be the cost of one of the newest PET scanners, medical grade ones. The one that was sent to Itemba back in the day, came from a London hospital, actually was acquired by, I think, by a pound. They were getting rid of it because it was no longer fit for purpose for medical research. Is the cost, it's not even the cost of transportation, but the cost of having all the infrastructure around it and the expertise. So yeah,
Okay.
I don't have a recent value for what a PET
Okay,
scanner
yeah.
would be.
But you also
But
need
there are
the,
many interesting developments. One of those would be, for example, combining a PET scanner with also CT, which some machines do now. So you can have both at the same time,
yeah.
and that opens a lot of possibilities as well.
But as you said, you also need to have the source near you. Yeah.
Yeah, exactly. And that's why, again,
Yeah.
it's not only about sourcing a PET scanner, but actually having a cyclotron that allows you to do that and all the expertise around that.
Yeah. Okay. Anybody else have question, comment, or? Okay. Let's thank Pablo for a very nice talk again. And excellent. Thanks for joining us today. Now you can have your breakfast and more coffee.
Catch a train. No, a pleasure. Thanks again for the invitation, George and Kevin. Greatly appreciated. It's been
Thank you
a
very much,
pleasure
Pablo.
talking to you all. And of course, if anyone has any questions later on, feel free to get in touch.
Yeah. And hopefully we can all catch up in end of October in Cape Town.
Yeah, looking forward to seeing you there.
Yeah.
Yep, sure. Okay, thanks again.